this video we will discuss about the clinical relevance of SETD2 and H2K36me3 levels in the human pancreatic cancer or human pancreatic adenocarcinoma. And this will be the last figure that we will discuss about this paper. And if you have any doubts, you can add in the comments or refer to the papers. Refer to the paper whose link has also been provided in the description. Let's begin discussing the results that are presented in this figure. So in order to find out this clinical relevance of this SETD2 and H2K36me3 levels in the human pancreatic adenocarcinoma, what I did is they check the samples of this human patients who are affected with pancreatic cancers, and using flow cytometer they find out the relationship between this cells that are rich in lipid and H2K36me3 plus tumor cells or tumor cells that have this H2K36me3 plus levels. And this is the strategy that they have used. First they separate the cells in this PDPN and EPCAM. PDPN is the marker for the CAF or cancer associated fibroblast, and thereafter in this cancer associated fibroblast they segregate the populations in this ABCA8A or FAB positive. And from the previous results we have identified that cells that have both these markers ABCA8A and FAB is represents this, both positive signals represents the lipid laden cancer associated fibroblast. And in the EPCAM they have identified this H3K36me3 plus positive cells in this forward side scatter wide and HK36me3 markers. This is the strategy that they have identified to check the percentage of lipid rich cells and H3K me3 plus positive cells. And they did it for multiple patients, around 43 patients, and what they have found out is that there is a negative correlations between this lipid laden cancer fibroblast and HK36me3 plus tumor cells. And we know that these lipid laden regions are formed when there is SETD2 knockdowns is there in the tumor cells. So this SETD2 has a negative relationship between with this H3K26me3 plus, and the quantifications are shown in this panel C also, where we can clearly see that lipid rich cells are high, and when the high lipid rich cells has low HK36me3 plus tumors positive cells. Low means less than 20%, and the reverse is also true, that is lipid rich cells are lower when the H3K36me3 plus positive cells are high, it is greater than 50%. And when they did the H&E staining, they find out one more thing, or they get further evidence to support their previous findings, that H3K36me3 plus low cells has more of this adipocyte like cells, that is lipid rich cells signals, and they are less differentiated, whereas H3K36me3 plus cells are higher differentiated, and they do not have, and no signals has been found for adipocyte like cells. And in the H3K36me3 plus lows, the triangles represents the adipocyte like cells, whereas there is no adipocyte like cells in the H3K36me3 plus high positive cells, and the cells are differentiated more in that case. And then they also did this staining of the H3K36me3 plus FAP and PDPN on this human pancreatic adenocarcinoma tissue sections, and what they have found out is that FAP has a negative relationship with this H3K36me3 plus expressions, and PDPN has a positive relationship with this H3K36me3 plus expressions, and these things are quantified on the bottom of the panel P, where we can see that the high H3K Me3 plus has high H3K36me3 plus cells has lower FAP, but when these receptors goes down, we see that FAP is high, and the reverse is shown for this PDPN. And after validating their results in the clinical relevance, they wanted to check whether this will work in vivo when the patient-derived xenograft is used for the for this human pancreatic adenocarcinoma disease in the mice. And they did a treatment after every other days from the 16th day. And what they have found out is that in the H3K36me3 plus low samples, they see a high impact of this S globin and JQ1 that can be seen here. P values are less than 0.001, 0.007 for the tumor mass, and for these are the two replicates. So for both the cases, P values are very low, and we can see that decrease in effect in the tumor volume is also very high. That is, when the mouse gets injected with this S globin and JQ1, they see a more tumor volume reductions. And similar experiment when they did it in the patient-derived xenograft of this H3K36me3 plus high cells, what they have found out the impact of S globin and JQ1 is not significant. It is P value is less than 0.032. P value is equal to 0.032 and 0.031, and also the decrease is not the effect on the tumor volume is also not that significant compared to the results that they have seen in the H3K36me3 plus low cells. And here they shows the results for the three different triplicates, and all the triplicates of this H3K36me3 plus high has P value is quite high compared to the H3K36me3 plus low cells. And also the effect P values for this tumor volume change is also high compared to H3K36me3 plus low tumor cells. So all this only validates their previous findings that H3K me3 plus 2 affect a lower amount of this H3K26me3 plus affect. Oh, this is typing mistake, it should be 36. H3K36me3 plus low will cause epigenetic changes, and that will result in the more proliferations of the cells, as lower amount of H3K26me3 plus will also in relationship with its counterpart, that is H3K27AC upregulations, and these upregulations will cause these upregulations of this H3K27A6 will cause epigenetic changes in a way that it will upregulate the genes that are involved in the proliferations and migrations. That is, this act as a suppressions of the genes, whereas H3K27AC act as a overexpressions of the genes that are involved in the that are involved in the proliferation and migration of the tumor cells. So I hope you understood their clinical relevance of the SETD2 and H2K36me3 levels in hPDAC. If you want to understand more about this topic or these experiments in detail, you can go through the reference that is provided in the description. That's all for this video. Thank you for your time.